Hello everyone and welcome to today's DDN webinar. My name is Yuning. I'm the Assistant Science Editor for DDN, and I'll be moderating our discussion. We have an exciting webinar planned for you today. Our speaker, Michael Breen, will discuss the role of RNA editing in neurodevelopmental disorders and how his team develops the site-directed RNA editing therapeutics. After the talk, Dr. Breen will participate in a live Q&A session. If you have any questions or comments during the presentation, you can simply submit them to the Q&A portal to the right of your screen. We'll try to get to as many of these as possible. I'd like to take this opportunity to thank our webinar sponsor, BPI's Bioscience. BPI's Bioscience strives to advance new scientific discoveries that lead to therapies by creating innovative solutions for research. BPI drives advancements in immunotherapy, epigenetics, coronavirus, cell signaling, and other research areas, providing more than 3,500 high-quality recombinant cell lines, virus-based tools, proteins, and biochemical and cell-based assay kits focused on drug discovery. BPS provides convenient screening and profiling services to identify potential new therapeutics with their in-house developed assay kits. As a primary manufacturer, BPS can supply products in large-scale bulk quantities as well as contract and custom manufacturing. Our sponsor has provided us with some helpful handouts related to today's webinar. You can find and access these in our handout section located on the right side of your screen, where you can also find your certificate of attendance for participating in today's live event. And with that, let me introduce our speaker, Dr. Michael Breen. Michael Breen is an assistant professor of psychiatry, genetics, and genomic sciences, and a faculty member of the Icahn School of Medicine of Mount Sinai. Breen earned a PhD in genomics and bioinformatics from the University of Southampton, United Kingdom, where he investigated epigenetic and transcriptomic mechanisms underlying a range of major neuropsychiatric disorders. He completed his postdoctoral training in molecular psychiatry and genomics at the Icahn School of Medicine at Mount Sinai. He specializes in human genetics and genomics, computational biology, and cellular neuroscience. His research group dissects cellular and molecular mechanisms that drive brain development and their dysfunction in neurodevelopmental disorders. Breen is also a member of the Severe Autism Center for Research and Treatment. Let's just take a minute to make sure your slides are working. Dr. Breen, please take the floor.
Super. Thank you very much for the introduction. I'm just going to jump right into it. And I'm going to start with one of the most important slides. So we're a lab that focuses on ADAR-RNA editing in rare disorders. And we're at the Icahn School of Medicine Mount Sinai. This is our campus. This is the building I'm calling in from today. Our lab is right about here, about the 14th floor. And I just wanted to give a big shout out to the members of the lab who give me a lot of motivation and inspiration in making it fun to come in to work every day and work on these problems. Okay, so today there are three areas of focus for discussion. So the first area we'll talk about, what is ADAR editing, why is it important, and how do we measure it? The second, we'll be learning rules of ADAR editing across developmental, cellular, and disease scales. And the third is we'll talk about how ADAR editing can be used as a therapeutic tool and how to measure its effect. So the first area of focus now. So ADAR editing, also abbreviated as through adenosine to inosine editing, right? So it's an RNA modification that introduces individual substitutions into RNA sequences, right? So here what I'm showing you here is a deamination reaction where adenosine is deaminated into inosine. Now, inosine is not a Watson-Crick pair, but it's a molecular mimic for guanosine during translation. So an A to I edit is read as an A to G change through this other machinery and by sequencing. So here's an example of an edited transcript, an unedited transcript, and an edited transcript. And so how do these conversions occur? There are ADAR enzymes that drive these conversions. So these ADAR enzymes have two major components. The first is a double-stranded RNA binding domain that bind to double-stranded pre-mRNA, and a deaminase domain that, again, catalyze these adenosine to inosine events. So what do these conversions, these basic conversions, do? Well, it really depends on their genic context, right? So if these conversions occur in the 3' UTR gene, it can affect the way that a microRNA might bind and regulate host expression. It can also have various effects near splice site regions where it affects alternative splicing, or encoding regions where it leads to a non-synonymous change that changes the way that the protein is translated into a different functional isoform. And there are a lot of different consequences and ways in which A2I editing can regulate gene expression and protein translation. And so one way to kind of understand where RNA editing and these ADAR conversions are taking place is just to ask, where are these ADAR enzymes expressed? And so there are three classes, ADAR 1, 2, and 3. And this is data from the Human Protein Atlas. And you can see that ADAR 1 is really broadly expressed across all tissues, whereas ADAR 2 and 3 are more selectively expressed in the CNS. ADAR 3 is not catalytically active, but it does have a double-stranded RNA binding domain. So it's thought as a negative regulator of RNA editing because it competes with the double-stranded RNA binding space with ADARs 1 and 2. But it's mainly, just to emphasize this, it's mainly ADAR 1 and 2 that are doing all the editing here. And so a big motivation for me to get involved in this field was through all of the work from the late Marie Oman, who has really laid a very strong foundation of RNA editing in the human CNS, or even the mammalian CNS. And this is a graphic from one of her review articles that really depicts the importance of ADAR editing around the neuronal synapses. And so perhaps the best studied site is this one here. And RNA editing in the brain was really initially observed in studying the main players of fast excitatory neurotransmission, so AMPA-GluA2 subunits. And so cloning of this subunit revealed this discrepancy between the genomic sequence and the encoded mRNA, where the genomic sequence had a glutamine, and then the mRNA sequence encoded was an arginine at a particular location in the coding sequence. And through deeper investigation, it turned out that having an arginine at this site really rendered this channel impermeable to calcium and decreased the single channel conductance of this activated ion channel approximately tenfold relative to an unedited version. So this particular site seems to be very important for regulating calcium permeability. But there are a number of other sites in neuronal synapses that seem to be important, not just for excitatory receptors, but also altering gating kinetics of inhibitory receptors, generating protein isoforms with different functionality for serotonergic receptors, altering circular RNA formations, etc. So again, there's a lot of ways in which ADAR editing can fine-tune gene expression and protein translation in the brain and around neuronal synapses. It turns out that some of these sites that have strong functional impacts also have different rates of editing at different periods in time. So here, if you take a stem cell or an NPC and you push it into being a mature neuron, the site that I discussed that is important for calcium regulation, the rate of non-synonymous change increases the older that cell gets, right? And so it starts off being very lowly edited, and the more mature this neuron becomes, the rate of exchange of this non-synonymous change occurring again becomes more frequent. And it turns out that there are a number of other sites that also have these temporal patterns that are very precise throughout neuronal maturation and differentiation that also have functional implications. And we'll discuss more of this in a bit. So how do you quantify A2I sites? So we are typically studying brain cell types or brain tissue, and we'll obviously RNA, we'll subject it to RNA sequencing, and we'll perform in silico analyses. And I won't discuss a lot of the methods for the specific in silico analyses, but I refer all folks to go through these for best practices and guidelines. This is a a number of different publications that have really done a good job in outlining those methods. But I do want to discuss two main modes of editing. So one is more what we would call selective editing, where you have your genome, and then you have your RNA sequencing reads, and perhaps at this location you found a number of aligned RNA reads that map to this location of the genome where the reference is different, right? So you have 4 reads here that are indicating there's a guanosine relative to a reference being adenosine, right? And so what we typically do for an individual site is measure an editing ratio. So you would look at the total number of Gs that are counted here over the total coverage of all bases at this location. And in this particular example, perhaps you have editing ratio of 20%. So this is selective editing, where you have an individual adenosine being edited typically within an allo element. And then you have RNA hyperediting, which is editing of consecutive adenosines along an extended cluster within the same transcript. And these are generally transcripts that are so edited that you have to fish them out from the unmapped reads from RNA-seq data. And again, I refer you to these papers for best practices for doing this. And I also just want to give a shout out to this method that is really widely used. It should be adopted more often as a gold standard. It's called the Allo Editing Index, and it's a really great way of summarizing global A2I editing activity across allo elements in the transcriptome. So allo elements undergo a lot of editing, and this is a way of effectively decompressing all of the editing activity across the transcriptome into one singular value. And serves for a bunch of different important purposes in your analyses. And it's also important because there's so many sites that you can discover. And so here's a plot of just some data that we've been analyzing recently. The colors and the labels are not so important here, but I do want to emphasize is that the number of sites that you can detect in a transcriptome sample is pretty dependent upon how deeply you sequence, right? So here we have some deeply sequenced samples from a cohort here in purple, and we're detecting upwards to 1 million A2I editing sites from those RNA sequencing data. So then how do you start figuring out which of these are functional? And this has been a main focus of research that we've been trying to do in terms of assigning sites functionality using data-driven approaches. So are they genetically regulated, tissue-specific, cell-specific, temporally regulated, et cetera, to build up an empirical database that might give priority or prioritization of sites to have more functional relevance. Okay, so this is a good segue into this next broader area of discussion of learning rules of A2I editing across developmental, cellular, and disease scales. So when you look at human brain development, there's clearly a lot of different functional phases that a brain goes through, right? And knowing what we knew about some of these important sites that regulate calcium permeability, having temporally regulated patterns, and other sites that are functionally relevant that also are precisely temporally regulated, we've recently asked the question, what happens across the transcriptome? What other sites might be temporally regulated? And so we started simply by computing that editing index, which again is a global summary of editing activity across the transcriptome, and asked, how does that change across prenatal versus postnatal periods of development? And here you have human dorsolateral prefrontal cortex from prenatal into postnatal periods. You see a pretty sharp jump in editing activity into postnatal periods of development. And this is in the dorsolateral prefrontal cortex. And you see a similar pattern in the whole forebrain or cerebrum, and as well as in the hindbrain or cerebellum. And these patterns and trends are actually echoed quite well in vitro models. If you push an NPC into a mature neuron, you can actually recapitulate these increases in allo editing across the entire transcriptome as a neuron matures. And so one way to summarize these effects here is visualized here. So you can take all these different models and studies and do a meta-analysis. And you can see that if you're on this side of the line, you're kind of increasing across time, or if you're here, you're decreasing. And this index of editing seems to be clearly increasing across time across these models, as is ADAR2 and not so much ADAR1. So we think a lot of this seems to be driven by editing activity of ADAR2 across brain development. We then pivoted, instead of looking at these global pictures of editing sites, we looked at the individual sites. And so here we're asking of individual sites, which ones differ the most between prenatal and postnatal periods of development. And so here you can see, if you're on this side of the plot, these are sites that are highly postnatally biased, that increase in editing levels across time. And these are prenatal, that actually decrease across development. And if you look at the breakdown, there's a lot that actually map to 3' UTR regions. So given the preponderance of these sites in 3' UTRs, we asked the follow-up question, what is the likelihood of these sites to be interfering with microRNA binding in these 3' UTR regions? And through our analysis, what we found was that actually editing on 3' UTRs during postnatal periods of development seemed to form more favorable structures for more stronger binding for microRNAs, and provided more substrates for microRNAs to bind during postnatal periods. So in other words, there is more editing in 3' UTRs during postnatal periods of development that once microRNAs bind, they form more stable bonds and can regulate gene expression to a greater degree. Okay. In addition to 3' UTRs, we also looked at these sites in coding regions. So these are RNA recoding events, meaning sites that will occur in protein coding sequences, but flip out and introduce non-synonymous changes, right? And so we found around 37 different RNA recoding sites that lead to different amino acid substitutions, and the rate of exchange of these substitutions increases generally across development. And so here are these 37 recoding sites in their respective genes. And here's just one good example. So this is, again, that classical site, this QR site in GRIA2 that regulates calcium permeability. And you can see during fetal periods of development, it's slowly edited and then reaches about 100% editing efficiency across all of these regions during postnatal periods of development. And there are a number of other sites that have precise temporal patterns with different maximum editing levels. This one reaches 100%, this one 20%, but doesn't have an effect in the hindbrain, but does in the cerebrum, etc. So there's a lot of lessons that can be learned about perhaps new functional recoding sites that could be studied in different ways that are important for neural development. We also asked, across fetal and postnatal development, what happens with these stretches of RNA hyperediting? We found many genes that undergo extensive hyperediting during advanced stages of aging. And here are kind of a list of all of these different genes that undergo lots of hyperediting. And then we measured, the effect of hyperediting on the double-stranded RNA secondary structure, here abbreviated as the minimum free energy structure. And you can see that if you're a gene that's more likely to undergo a lot of editing, your natural minimum free energy is generally a little higher than those that don't undergo a lot of editing. And that after editing during postnatal periods of development, this change is more pronounced this change in minimum free energy is more pronounced, right? So in other words, genes that undergo extensive hyperediting during postnatal periods of development typically show more stable secondary structures. Okay, and then the next thing that we wanted to look at, and one thing I think is important to emphasize, is that previous work from us and many other groups have looked at ADI editing levels that are disrupted in different diseases. And we can find lots of different individual sites where editing patterns are changing in, for example, schizophrenia compared to controls, or autism compared to controls. And so we can take these sites that we find to be, differentially regulated and ask what their developmental relevance is. And before I move on to that next slide, I just want to emphasize that even in this example where you have two independent studies of schizophrenia and autism, you find sites that are significantly different and seem to map to similar types of functional processes, mapping to, genes that are implicated in glutamate receptor activity and ion channel complexes. So you can take, these sites and you can ask, hey, are these sites also temporally regulated? Do they have to be edited at particular levels during different periods in time? And so we know that the mean editing rates for thousands of sites seem to look like this over development. And we just ask, what fraction of these seem to be disease related? And we do see, a general enrichment for autism and schizophrenia dysregulated sites to also be temporally regulated and increase in editing levels in normal brain development. So this is another way of showing that, disease-related sites also are required for, throughout normal human brain development. So a lot of this work has been performed on bulk tissue RNA sequencing. One way that we try to get at the cellular diversity is through this method called fluorescence-activated nuclei sorting. And so here we'll take a brain tissue of interest and use an antibody-based approach, so NUN or RBFOX3, to sort out neuronal versus non-neuronal cells. And then you can add SOX6 to pull out GABAergic from glutamatergic neurons, and then SOX10 to pull out oligodendrocytes from microglia and astrocytes, and then IRF5 to isolate microglia versus astrocytes through the SPANS methodology. And so the benefit of this is you can sequence very deeply these targeted pools of cell types, and you can study editing within these cells. And so here's an example where there are 5 different cortical areas where the NUN antibody was used to pull out neuronal versus non-neuronal cell types. And you can see that the rate of editing computed by the L editing index indicates that editing levels are generally much higher in neurons compared to non-neurons, with not a lot of regional specificity. You can add another antibody, so to the NUN positive fraction, so the neural positive fraction, to pull out excitatory neurons. And you can see that indeed GABAergic neurons here seem to display an elevated level of editing activity relative to glutamatergic neurons and oligodendrocytes. And you can add another one here, antibody here, to pull out the microglia and astrocyte population, and you see a trend like this where, again, highest editing levels in GABA, followed by glutamatergic neurons, microglia and astrocytes, and then oligodendrocytes being the lowest. Given those trends showing that neurons generally have a higher level of editing, you can actually take a bulk tissue sample and ask what fraction of editing variance in that sample might be explained by neuronal content. And so here we show that across several hundred postmortem bulk RNA sequencing samples, that the rate of editing can be somewhat predicted by the fraction of neurons within that particular sample. And then another way that RNA editing can be regulated is through common genetic variation. And so one of the ways that we started looking at this was we first noticed that most sites that are edited are not generally edited across large amounts of samples. So virtually all adenosines and allos can be edited, but are typically consecutively edited across hundreds of individuals. And so one way to kind of start piecing that out is at least coming from a human genetics background, was, are there any underlying genetics that might explain variability in editing? And so this is where the kind of the cis editing QTL analysis comes in, these editing quantitative trait loci, where you'd ask, does a local SNP have an effect on editing levels? And when you do that, you'll find thousands of editing sites that are tightly associated with the genotype of a local variant. And these SNPs are generally located in cysts very close by. Here's a majority of SNPs that you find to be regulating editing sites. And this gray window is 200 kb plus or minus, but if you look closer, it's really around 5 kb. And here's just an example where the editing level for a particular site can be explained by whether you're homozygous for the reference or alternatively or heterozygous. And these are pretty pronounced differences, right? So moving from 20% up to 70%, 75% editing levels for one particular site here based on a common genotype. These genotypes can also vary across brain development, which I think is pretty important. So you can look at the effect of SNPs on editing levels in prenatal periods versus those effects on SNPs during postnatal periods. And you see there's a strong kind of prenatal predominant effect that's absent in postnatal periods. And here's an example. So if you're looking here, you're in prenatal periods, and here you're in postnatal periods. So this is a SNP. Green is your homozygous for the reference, blue is heterozygous, and purple is homozygous for alternative allele. You have a very clear separation in editing levels prenatally that then wash out as you age and into postnatal periods of development. So these are examples of prenatal specific QTLs or context specific editing QTLs. And one way that you can start, making sense of these, at least in light of complex disorders and traits, is by studying if there are any genetic risk variants that might perturb A2I editing levels. So this is from a study that we did a few years back where we looked at the rate of co-localization of disease risk variants to co-localize with editing QTLs. So let me walk you through this. So here's a GWAS signal for schizophrenia. And here's the SNP for that. And then here's the same SNP that also has a strong QTL effect. So the SNP is also known to have a strong relationship with the local editing site and its editing levels. So it's a way of finding an intermediate molecular phenotype. Here, the editing levels perhaps being a causal path for disease pathology. Okay, so the third area of focus, we'll move on to there. So the third area of focus is, how is A2I editing being used as a therapeutic tool, and how to measure its effect. And so there are a number of different ways now to leverage endogenous ADAR enzymes to repair G to A nonsense mutations. And so here's a general schematic that I'll walk you through. So perhaps you have a disease variant of interest that causes a nonsense stop mutation, right? And so you design these guide RNAs that are complement to your mutated region of interest that will recruit, once they hybridize to the region of interest, endogenous ADAR1 and 2 enzymes. And after it hybridizes and ADAR is recruited, it will edit this TAG stop codon back to, in this case, a tryptophan for mRNA and protein read-through, right? So it will abolish the nonsense mutation and allow the protein to read through. That's the general idea behind recruiting endogenous ADARs to repair G to A nonsense mutations. And so what does the landscape or spectrum of G to A mutations look like that might be suitable for this type of therapeutic approach? So here we've taken HGMD, which is a very large, well-curated database of disease-causing mutations in humans. And total, there's around 115,000 disease-causing mutations. We will filter those down to only G to A variants, right? Because we want to flip them back to healthy alleles, and we can only edit A to G. And so then we're down to about 20%. So 20% of all of these disease-causing variants are G to A mutations in this database. And you see that there's a large fraction of them that are actually missense and nonsense. You can ask further, you put further criteria onto these variants. You can ask which of them have high levels of conservation and pathogenicity and anything that has a high level of pathogenicity conservation is closer to 1 here. So you put a threshold on that. And then you can ask which of these remaining variants overlap with genes that are, whatever your specialty and question of interest. So we're studying developmental disorders. We might overlap them with a range of genes that we know are already linked to autism and other neurodevelopmental disorders. So we can go down to, 1,500 variants. 75% of them are nonsense. We can then start ranking them by optimal mutation codons, and those that are expressed in the most disease-relevant cell models, to start getting into the best candidates for us to think about therapeutic repair. And so here I'll just outline 2 ways of recruiting endogenous ADAR enzymes that have been proposed very, very recently, but have really created a lot of enthusiasm in the field for doing these types of therapeutics. And so one is this approach called Restore, where you have engineered this short, chemically optimized oligonucleotides that have a specificity domain and an ADAR recruiting domain. This is a hairpin structure that recruits ADAR, and it's not a plasmid-borne approach. And again, it's chemically modified. And there's another approach here, which effectively will just design, called LEAPR, which effectively is really longer linear oligonucleotides of 100 to 150 base pairs, no chemical modifications. And it will recruit ADAR effectively through the formation of this duplex. When it binds and hybridizes to the region of interest, ADAR is recruited. And it can be used as a plasmid-borne approach. And then a third approach that we've also been using is this work that's been done very elegantly by Prashant Mali's lab, where you can actually circularize these guide RNAs with twister ribozymes. So here you can add these ribozymes upstream and downstream these ligation sequences to circularize your RNA. You can see that generally these circular RNAs on this target of interest, 3' UTR of RAP7A, seems to lead to higher editing efficiency relative to the linear counterparts. And so one thing that we've been using to measure RNA editing efficiency and different guide constructs and trying to design useful guides to repair mutations is this reporter line. So we have effectively these hex cells that are expressing a broken GFP. So we have enhanced GFP harboring an in-frame UAG stop codon that will only fluoresce after ADA-I correction to a UIG, right? And so we can test different linear and circular guide designs. We can, and only after correction does this reporter turn green. We can put this into an IncuSite and do live cell imaging over extended hours. And so here's a little bit of insight into some of the results that we're getting. So here's a linear guide at 150 base pairs versus a circular guide. And you can see that we're doing live cell imaging for GFP at 12, 24, all the way to 96 hours, where this circular guide seems to be leading to enhanced GFP expression over time relative to its linear counterpart. And they seem to just be more stable, right? And hardy over longer periods of time relative to linear guides. Okay. So this was kind of a tour of many different things that we're doing. This is a little bit of a preview into different ways of using reporters to study mutations of interest. And here's a brief summary of things that we've went over. So, ADAR editing is really quite an abundant modification that's driven by ADAR enzymes. In coding regions, it seems to be important for regulating synaptic transmission and neuronal signaling. And you also see changes over time, right? There are thousands of sites that are temporally regulated, not just those in coding regions, but also in 3' UTRs, hyper editing as well. And as you move throughout kind of refining your focus, there's a lot of editing more in neurons compared to non-neurons. Common genetic variation also seems to play in regulating editing levels. And then, using insights from these studies to move into therapeutic approaches to find ways of repairing G to A variants using endogenous ADAR enzymes has been a big area of focus for us, but also many others right now. It's a really exciting time to be part of this field. With that, this is the concluding slide. Again, this is our lab, and I'm just really proud to be part of it and to be working with these people. If anyone's interested in RNA computational biology, we're always looking to collaborate or hire. So thank you.
Thank you so much for the presentation, Dr. Breen. In the time we have left, let's get to our listeners' questions. So first question, I'm wondering, why do neurons or the brain have a much higher level of RNA editing compared to other tissues? Do we have any explanations for that?
Yeah, so a lot of the times where you find, so it's a simple question, but there's a very long answer, but I'll try to give a short one. There, typically in the brain, a lot of the editing that we see is driven by ADAR2 expression and activation, right? And so there are some cell types that express higher levels of ADAR2. And this seems to lead to elevated levels of editing within those populations. There are many other factors that you need to consider outside of ADAR expression, is where I'm trying to go with this presentation, that explain RNA editing variability. The developmental specificity, clearly if you're an older sample, there's a lot of editing versus if you're a much younger sample. The genetic component of it. I didn't even get into RNA binding proteins, because there's many other RNA binding proteins that also have double-stranded RNA binding domains that can compete with ADAR binding and also be negative regulators of editing activity. So there's many factors that really lead to elevated editing in neurons and in brain tissue.
Thank you. We have a question from Tomohiro. What do you think causes higher editing efficiency by circular gRNA?
Yeah, I think it's been kind of generally accepted as a community now that these circular guides are generally more stable than their linear counterparts, and as a consequence are not susceptible to degradation very quickly. And so they stick around longer, and there's higher editing levels. That's what we've been working off of.
Thanks. You also mentioned the rate of our editing as also the level of editing. I'm just wondering, what's the difference between editing rate and editing level?
Yeah. Sorry if I lost anyone with that. So I use them synonymously. Okay. The level of editing, is really a metric from 0 to 100%, right? Where you're generally looking at the fraction of edited reads versus total coverage of reads piling up at a particular site. So if you're 0%, you're not edited, or if you're 100%, you're all the way edited. And so, okay. So editing level is where I'm going with that.
Okay, thanks. You also mentioned that you notice like hyper editing during aging, and the RNA structures are more stabilized. Do you know why?
Yeah, so we're dissecting this in a few other studies. I didn't present any preliminary data or unpublished work here, but we think a lot of it has to do with inflammation and inflammatory processes during later stages of development, specifically in individuals over the age of 70, 80 years old. And so that's kind of our assumption that, I did talk a lot about ADAR 1 and 2 driving a lot of editing. One thing I didn't emphasize is that ADAR1 itself is also tightly involved, and one of its core functions is regulating the innate immune response and inflammation by editing double-stranded RNAs. And so we think that there's likely another mechanism there by which ADAR1 is responding to inflammatory processes during later stages in development.
Thank you. We have a question from Sanjay. So what might be the function of such high levels of allo repeat editing, and what is ADAR3, I think, doing being specific to the brain?
Yeah. So so the first part of that question, what's the relevance of editing in ALUs? So ALUs are primate-specific repeats. They seem to harbor a lot of editing and inverted repeats. So they form they end up having these like palindromic sequences that form these double-stranded RNA structures that undergo a lot of editing. And so ALUs themselves are generally fairly densely edited, commonly edited. We see them not just in humans, but in our work, and I didn't present it, but we also see it in non-human primates and macaque and also other mammalian models of development like mice. But mice is clearly there's reduced levels of editing compared to non-human primates and humans. And then ADAR3, and that's a good question. So ADAR3 itself in the brain is really, from what we see, restricted to a subset of inhibitory cells, inhibitory neurons. And, we haven't really deeply looked at ADAR3 abundant cells in a deep way, but I think it should be a focus of future work. But typically, a lot of the major neuronal and non-neuronal cell types are expressing lower levels of ADAR3, but ADAR3 is really quite high and abundant in a small subset of inhibitory neurons.
Thank you. You also talked about you investigated the genetic risk variants in neurodevelopmental disorders that can disturb RNA editing. Can you tell us a little bit more of that research? What findings do you have for this line of research?
Yeah. So there's been a lot of progress, not just by our group over the years, but by others. And so I'll talk mainly touch on mainly the work from our group. And so we've been looking at how common disease risk variants might also perturb the level of RNA editing. We've been specifically asking questions historically, by the nature of our research, about what fraction of disease risk variants for neurological disorders impact A to I editing levels. It's an interesting question, I think, because a lot of the disease risk variants lie in non-coding areas and don't typically have a expression QTL assigned to it. So we're looking for other ways to explain, pathogenicity of a SNP or an intermediate molecular phenotype that can be studied in a deeper way. Our work has touched on schizophrenia, bipolar, sleeping disorders, and OCD. But it's an ongoing active area of research, and I think it's still very young. But I think if there are ways to start therapeutically identifying SNPs that perturb critical RNA editing sites, those represent interesting candidates for therapeutics.
Thank you. We just have another question coming in from Tomohiro. Are the dsRNA binding region structures the same for all of the ADAR subtypes?
Sorry, if I understand the question, it's if the binding regions are the same across ADAR 1, 2, and 3, or different?
Yes, I think that's the question. Yeah.
Yeah.
Or
So
the dsRNA.
so let's focus on ADAR1 and 2. ADAR1 is really, it's known to be an editor in chief of long double-stranded RNA secondary structures. ADAR2 does a lot of a critical protein recoding and coding sequences. Although there is recent work showing that if there's a deficiency or lack of ADAR2, or in some cases ADAR1 might come in and edit ADAR2 sites. Okay, ADAR3, doesn't edit at all, but it compete for binding space, which is, I think, to your point of question. There will be a lot of work coming out over the next few years showing, using eClip or ClipSeq data, binding footprints across different cell types and tissues of ADAR1, 2, and 3. This hasn't really been done in a very detailed way yet. There are some studies, but clearly not enough to fully address that question. But it's definitely a way the field should be progressing right now. And I know it is moving in that direction.
Thank you. Can you also comment on the challenges of targeting ADAR RNA editing to develop therapies?
Yeah, I mean, which one? I mean, there's going to be a lot. I think my stance on it is a little bit different from a lot of the ways folks typically get involved in site-directed RNA editing approaches. A lot of individuals start with, a gene of interest and go from there. I think there's a lot of utility in trying to be a little bit more unbiased and trying to start with a landscape of mutations and dwindling them down based on important features that can be experimentally tractable and positive. So there is a lot of work on disorders of the liver, which have high uptake of AAVs if you're going to be, putting these guys into an AAV for delivery. There's some research ongoing with MICP2, which itself is a challenging gene to work with in other ways. There's a lot of gene-specific factors that I think make, each target unique and challenging in its own ways. And I think we don't really know what those challenges are until we start going after them. And unfortunately, there are a lot of them are so sequence intrinsic that make, for example, binding probable. Is the site accessible? Is the gene expressed enough, et cetera? So we don't really have a rubric right now to really kind of prioritize variants, but there are a lot of obstacles certainly in developing an RNA therapeutic, but also any therapeutic. But I've been very encouraged by the progress on individual candidates thus far across different labs and companies.
Thanks. And now we have time for one more question.
Sure.
Dr. Breen, just tell us about what's next for your research.
Yeah, so I think we kind of touched a little bit on some of it. So we're really quite active in two areas right now. One, thinking about different ways that common variants could be useful in therapeutically in targeting therapeutics for different diseases. So with the co-localization stuff, but also what does it mean for double-stranded RNA secondary structures? We're also moving into an area of RNA therapeutics for monogenic disorders, like a lot of people are right now. But again, we're taking an approach that is probably a little bit different from most in starting with targets that we think have the highest probability of success.
Thank you. Unfortunately, that's all the time we have for today. If you have any further questions, please consider reaching out to the speaker directly. His email is shown on the screen. As a reminder, the webinar will be archived on the DDN website, and you will receive an email notifying you when the webinar is available on demand. On behalf of DDN, I would like to thank our speaker, Michael Breen, as well as our sponsor, BPS Bioscience, and of course, our thanks to you for listening. Goodbye and have a good rest of the day.